My name is Damon, and our cameraman today is Michael.
My name is Jason. I'll be playing the Brazilian Rancher.
I'm Nathan. I'm geneticist.
Afternoon.
You bet. So Brazil is the largest commercial herd in the world at around 225 million head, and only about 20% of that gets exported. We're looking to import some into the States to increase that a little bit, and I was just curious, what you guys are running.
Yeah, we run a lot of cattle. We run these Neolord cattle. They're Boss Indicus type. They really fit our environment well. So we like them, and we can't really change from that. We want to send you guys a lot of product, and I know we're not quite getting it there. We'd like to improve on that. So what we need to do.
You bet. So the biggest issue right now with these cattle, the tenderness and the marbling isn't there. So your meat's looking a lot like that. And then what we'd like to do is select animals based on their RNA and be able to add more marbling, a more favorable eating experience with each of that with the animals.
Yeah, I think we can do that. I think we should. How are we gonna go about doing that? What's up?
Yep. So like I said, we'll be able to select animals based on their RNA sequences, and Damon will tell us a little more about that.
To get the complete sequence of RNA, you have to go through the central dogma of biology. The first step is to transcribe your DNA into RNA. It's done in the nucleus, as you can see over in the cell, and the RNA polymerase, which is this molecule outlined here, starts it all off. The RNA polymerase attaches to the promoter, and the Tata box shows or tells the RNA polymerase where to start. It's called the Tata box because it contains adenine and thymine nitrogenous bases. The RNA polymerase copies the DNA strand downstream from the Tata box. Technically it should be over here, and while it's copying it, it's also re-zipping up the DNA strand. It keeps transcribing until it gets to the termination sequence, and then that's when it tells it to quit transcribing. The RNA polymerase tells it that. After you get your sequence, as you can see here, the 5' end gets a cap. It's also called a 7' methylguanosine to help initiate translation, and the 3' end gets a poly-A tail of adenine bases, usually about 50 to 200 bases, which gets added for stabilization. The next is to cut out all the introns and leave the exons, because we all know the exons are expressed. And now you have a fully functional RNA unit ready to sequence. And the way they sequence that is once you get your template, you isolate the template, and once you get what you want, you sequence the fragments, align on reference, and that is what they use to figure out which animals have the RNA sequences desired for tenderness, meat quality, marbling, and they select off of the RNA sequence. And this was the article we used to figure out all of our information for this project.